Good afternoon. Thank you so much for the invitation to present to you all. Thank you, SCIEX, for hosting this symposium. I'm going to be talking to you about PFAS in food, the various human exposure pathways, and some of the grand challenges. And so my name is Cheryl Murphy, and I am the director for the Center for PFAS Research at Michigan State University. So as you all know, you probably already know what and where PFAS are. Um, when we're talking about PFAS, we're talking about these per and polyfluoroalphalytic substances. They're characterized by having a carbon backbone with fluorine attached to the carbon. These are two of the compounds that we mostly talk about, we know more about. PFO and PFAS, they have 8 carbons. But there's other types of PFAS out there, such as Gen X, which have a shorter chain, and then there's some that are polyfluorinated as well. There's estimated that there's more than 9,000, maybe even 12,000 of these compounds out there. They have a hydrophilic part to them, and so as a result, they like to take rides on water, and they like to hang out at interfaces in the environment. Um, they're easily transported through the water and air and surface water. And because they usually hitch rides on water, they're usually easily taken up by plants, and they also go into animals, including fish and livestock. These compounds are known to be very remarkably resistant to degradation. It takes a really lot of energy to destroy that bond between the carbon and the fluorine. So as a result, these products really don't break down, and there's no really there has not been identified any type of biological mechanism that can break this in natural systems. And since they don't break down, and we've been using them since the 40s, 50s, they're starting to build up in our environment. We find them in our drinking water, we find them in our foods, we find them in industrial, military, and consumer products. And you can see here on this slide, there's quite a few of these products that they're found in, such as wrapping, like popcorn bags. We have them in dental floss. We have them in firefighting foams and makeup and ski wax and guitar strings, etc. They're also found in carpeting, and it's a concern because of babies that crawl along on carpets and then put their hands in their mouth. It's found that what we're finding is all across the globe that these PFAS molecules are found at measurable levels in almost every single human that's been tested. Where are they found in the environment? Well, they're usually found close to manufacturing sites or military bases or airports. And right before COVID-19, this was beginning to become the biggest environmental issue. This is a map put forth by the Environmental Working Group, and they update it periodically, but basically they're showing where the sites of contamination has been found in water across the US. And you can see that there's some hot spots. For example, where we live in Michigan, you see that there's quite a lot of PFAS sites. You see some more on the East Coast as well. But what's really remarkable about this map is that you see some blank spots, and we know that these chemicals do not stop at borders. And so really what this is reflecting in this map are areas where people are not looking for PFAS yet. Michigan was pretty active, and so they went out and aggressively looked for PFAS, and they found it. There's still quite a few states that are not looking for it yet. The effects on human health are numerous. It seems to be called what we call a systemic toxicant, or a general messer-upper, pretty much. It seems to affect most of our systems. And that has to do with the nature of the compound itself. It has this hydrophobic and a hydrophilic part to it. And this means that in biology, what do we have that has that same properties? Are cell membranes. So these PFAS molecules find themselves at home in our cell membranes. They also like to bind to proteins. And some of the most sensitive endpoints that have been found in humans are usually related to immunofunction or immune function. But they've been linked to kidney cancer, liver cancer, reduced response to vaccines, a lot of metabolic disorders as well. But it's hard to generalize the effects. It seems that there are sex and species differences in uptake and elimination. And so it makes it hard to generalize what these chemicals do in us and in a lot of other taxa. Of concern is, and I think that's what's driving the recent regulation thrust, is the PFAS impacts on children. And we know that there's been some evidence to suggest that it impacts immune function. So basically you have reduced vaccine efficiency, increased immune-related disorders, asthma, allergy, colds, and then suppression of antibodies, etc. And then there's other things like autoimmune disease that seem to be sort of correlated as well. But also, too, they're finding that kids that have been exposed to PFAS also have these metabolic signatures occurring. So it's affecting the way we are growing and developing. How does it get into food? Well, let's think about the PFAS contamination in the agricultural sector. So basically, this life cycle of it essentially. Basically, it's used in manufacturing, and sometimes the waste are landfilled or they're just dumped into the wastewater treatment plant. And then basically wastewater treatment plants will take it up and then they'll have it within their system. They're not filtering it out. So sometimes it'll go into the sludge or it'll go right back out into the water. Also, the things that go into the landfills eventually will leach out and will contaminated groundwater. In our homes, PFAS goes down the drain and into the trash. We have a lot of PFAS compounds that we use in our homes, and it goes down, or it can go into the water, again, into the wastewater treatment plant. So it can either end up into the environment where it gets into our fish, or the biosolids might actually be applied to farm lands, and then it's applied to as a field, a biosolids field, and then what will happen is, it'll go up into the plants, and then any animals that eat those plants will also accumulate PFAS. So we get it from a lot of different sources, either through drinking water or from our diet. And it's estimated that maybe 5 to 10% of our cropland has these contaminated biosolids applied to it. So that's quite a bit. We can get it from our water. There's also some legacy pesticide applications that might have had PFAS in them, and especially the containers that house them. And we also might get some contamination from packaging materials. So back in 2017, 2018, the state of Michigan decided to really look for PFAS across its state. So they formed a united agency. They united all their agencies. They formed this thing called Michigan PFAS Action Response Team. So they united Department of Health and Human Services, Department of Ag, Department of Environment, Great Lakes and Energy, Michigan Department of Natural Resources, etc. They all, and transportation, they all united under this umbrella of the PFAS Action Response Team, and they aggressively went out to try to look for PFAS in the drinking water, and they identified a number of different sources. So this is just a Michigan specific one, but I'm sure there's a lot of other states that probably have similar stories to tell if they start looking for contamination. But in Michigan especially, 45% of our citizens are served by groundwater. And then we have a public water supply also uses groundwater, which serves another 1.7 million Michigan residents. And so basically there was some concern, like in 2016, the EPA had issued advisories of 70 parts per trillion. But basically the state said that was too high, and they lowered it to 6 to 420 parts for some range of PFAS. And now there's some suggestions that the guidelines should be at 4 parts per trillion, which is essentially at detection limit, right? So they're saying that really we shouldn't have PFAS at all. And so Michigan went out to look for it, and they identified several sites that had PFAS in them, in the surface water, and in the groundwater. And it's of concern because of the environmental persistence of PFAS. So I said they don't break down, they persist in the environment. And because they don't break down, they have a tendency to bioaccumulate because they bind to our proteins and then they just stay there and they're not really eliminated easily, or metabolized, broken down. Um, and and as they accumulate, they can be toxic at parts per trillion levels. And then this is just a table to show you how long and they all have different half-lives. And this is just to show you how long they stay in our body. So the estimated half-life would mean how long it takes for half of the concentration that's in your body to go away. And so I think the one that's in concern is, like, for example, PFAS has a really long half-life. It takes 3.4 to 5 years for you to get rid of half of it. That's that's saying that as long as you have clean water and clean food, it'll take 5 years to get rid of half of it. But that we don't really have all that clean water, clean food, and that means like that 5 years is 1,826 days. And so if we have PFAS in our water, and this is where this parts per trillion things, we become thousandfold more contaminated with PFAS than the water we drink just because we bioaccumulate it and it stays in us and it doesn't break down. And so that's why they have to put these really low levels of in the toxic parts per trillion on the advisories. So how does it all get to humans again, just to recap? So for the majority of the human population, most of our exposure to PFAS comes through our food, right? So that's of concern. So most of us get it through our diet. And what we found in the general population is that the levels of PFAS and PFOA in the blood are about 100 to 1,000 times fold higher than what we find even in the drinking water. Okay, so that's coming from our food and also what's in our drinking water, but it's because it's accumulating, and not breaking down, and it has a really long half-life. When drinking water is a problem, it becomes the most dominant contributor to the PFAS in our blood when we live near contaminated sites like industrial and military. But there's still, like, we're just figuring this out, and there's still, like you saw, like there's a lot of the US that hasn't even started testing for it. And so we have a lot of knowledge gaps, which means who are the more vulnerable populations. And so this is an area that needs to be discovered. So like I said, it is getting into our food. So in the natural systems, it's getting into our fish. So this is just an example of some eat safe fish guidelines that Michigan DNR put up. And you can see that in areas like, for the Au River, for example, they say that you should not eat any suckers or any sunfish at all, ever. And then there's some limitations of what you should do with uh or rock bass or smallmouth bass, but there's limitations on how much rainbow trout. Let's see, what else do we have? Bluegill. So anything that has like a bluegill that has PFAS, they say don't eat. Largemouth bass, you shouldn't eat any of it. Rock bass don't eat. Smallmouth bass don't eat. And so what we're seeing too is what's weird about the PFAS problem is it's accumulating in fish species that would not normally accumulate contaminants according to how we know contaminants. Like, it's usually accumulating in the biggest and the oldest fish, and it's just not doing that. It's showing up in like sunfish and things like that. So yeah, and so it shows a very different pattern of accumulation than the other contaminants, and it's causing shutdowns in our fish. So there's a few streams and rivers in Michigan where you should not eat it, and also you shouldn't eat deer. Like, this is within a 3-mile radius of Oscoda, which is a site of the Wurtsmith Air Force Base. They're finding really contaminated deer as well. So so for hunters that eat wild game, this is a source of contamination. But it's also showing up in our farms too. So this is a farm that's located near MSU. They put biosolids from a wastewater treatment plant that accepted wastewater from industrial sources like chrome plating, and it was full of PFAS. And he used these biosolids on his farm for years, and went into his crops and into his cows, and so he was shut down. So there's lots of unknowns. So, I mean, we're starting to get a little bit of the idea of where it's going into our food supply and our trophic food web. But there's still lots that we don't know. We don't know all of the exposure pathways. We don't know how it biomagnifies in the food web because we see weird patterns. It's not showing up in the top predators. It's showing up in these like middle, like bluegills and things like that, and smell. Why? We don't know. We don't know what it's doing to the actual animals out there. And as a result, we don't really have a good handle on risk. So for example, to assess risk, we need to know what the exposure pathways are. And we also know once they're exposed, we have to understand what that does to the organism. And then the combination of these things is how we categorize risk. We're starting to really make a lot of effort on trying to figure out how it's moving through our environment, but there's still lots that we don't know. And there's very little known about what it's doing to us, especially all of these other compounds. Like we're getting a handle on PFAS and PFOA, but there's thousands more that we really don't know what they do, and we can't seem to generalize. So as a result, with this big problem, this is a complex problem. So at MSU, in response to the state forming their MPART, we decided, well, we should do something as well as a land grant institution. We should do something to help the state. What are some of the things that we could tackle as a research institution to help? And so we identified some of these big needs and opportunities. We knew that we have to characterize PFAS in the environment and the food supply. What chemicals are there and how much? How does it contribute to health effects, especially with multiple stressors? These compounds aren't occurring by themselves. They're usually occurring as complex mixtures of PFAS, but also with mercury, BCBs, DDT, and PAHs, but also temperature changes and things like that. So it makes it hard to tease out the effects of these compounds. How does it transport and accumulate through the water, the environment, and the food? And how do we get rid of it and replace it? And of course, one of the most important parts is how do we communicate the risk and formulate policy with all this uncertainty? This has to be done really carefully so that we don't like oversell things. We have to make sure that we're very careful about what's happening so that we don't lose trust. So we formed a center as a result of all of this. Because we wanted to have this multidisciplinary team to take on this very complex interdisciplinary problem. We wanted it to be comprehensive outlook and solutions base. We wanted a cohesive unit so that we could interact with state and federal partners. Each one of us, excuse me, could all go out and have individual grants, but we thought we'd be more effective if we were working together and like working with our state and federal partners a little bit more cohesively. And this is exactly what a land grant institution do. And because we have this land grant institution, it has a very well developed outreach programs. And so we have a conduit to our community members. And so this is how we formed our center. We identified 6 main research bubbles that we thought we could tackle. And we became a center that was university-wide. We have 9 colleges and the Fraunhofer Institute, all represented with about 30 or so and increasing members. The middle here, which you at SCIEX will be interested, we need to figure out how to measure all these thousands of chemicals in the millions of matrices that are out there. And we have to do it a little bit more efficiently and cheaper than we've doing. So there's lots of areas to be researched here, which we've developed an analytical lab to start to tackle that, and would love to collaborate with people on this. We have to monitor and model, like, where is it occurring in the environment and in people and fish, and model its transport so we can start to get some sort of predictive capabilities built in. What are the health impacts? What is it doing to us? We know we're starting to figure out two chemicals, but what about the thousands? And how does it affect fish, wildlife, as well as humans? So we have thousands of millions of species here. We need to move away from like the traditional way of doing toxicology and move into more systems talks, I think, to tackle this. Because we're an agricultural college, we thought, how do we keep our food supply safe? What is our strategies? We should develop some solutions on developing food safety strategies to keep our food supply safe. How do we assess the risk and communicate it effectively? We need to do this very carefully. And how do we manage it? How do we clean it up? And how do we develop safer alternatives? So I'm just going to talk about some of these research bubbles now. So looking at the environmental analysis and chemical properties. So we know we have about standards developed for about 40 of these PFAS using, there's an EPA draft method 1633 that measures quite a few of them. These methods developed for drinking water, for EPA 537 and 533. We have some FDA methods for food, but, we have lots of other chemicals to measure. But we also need standard reference materials, and NIST has some of them, but this is it's a reference, it's a it's a it's a collection that needs a lot more because we only have a few PFAS and a few matrices, so we're missing standards for the vast majority of PFAS chemicals and in the thousands of matrices. So there's lots that has to be done in this sphere. For monitoring and modeling, so we've monitored and modeled like two of these compounds, PFAS and PFOA, quite extensively, and there's a handful of other molecules that are measured occasionally. But the transport through the water is just now being modeled. And so just for two of these chemicals, so there's lots that has to be done here. Some states monitor and regulate PFAS levels in biosolids. So that's starting. There are a few TKT models for model species, but we have to look at, most of them are complex mixtures, and we have to figure out what they're doing. And oftentimes, PFAS will compete with other PFAS molecules in biology and boot it out, but we don't really know how all of it works together. And we have thousands of other chemicals to monitor. And in this research bubble, the health impacts and food safety. So we talked about how the health impacts are widespread and varied, but we really need generalizable patterns. We're starting to see some regulatory thresholds for foods. So for example, in the European Union, you have some for eggs and fish muscle and bovine, pig, and chicken meat at fairly low levels than what we would see sometimes. There's been only regulations proposed for very few PFAS. So we have PFOA, PFAS, and a limited mixture here in the US, and that still hasn't come out yet. It's been proposed and hopefully comes to become a regulation soon. But we also make assumptions that ingested food follows the same dynamics as PFAS in drinking water. But you know what? The tissue concentrations of PFAS within an organism vary from organ to organ, and even within the matrices itself, it's not very homogeneous. And we have thousands more to study, and we really need to move to some more high throughput new approaches and modeling. Now, the risk assessment and communication is incredibly challenging, like I said, because of the uncertainty in the number of chemicals and the environment and their accumulation and toxicity potential and interactions and mixtures. There's been some suggestions that we should regulate as a class, but then there's a lot of pushback on that by a bunch of different groups. So this is not resolved in any state in any way. We need to be able to communicate risk with some known solutions, actually, because otherwise the problem will just get buried and we won't do anything about it. And we have to develop best strategies. And I'm just going to give you this example, of this guidance on PFAS testing. One member of our center worked on a panel with the National Academy of 16 panel members for 2 years to come up with the best guidance for PFAS testing for physicians. And so that took a lot of careful thought and work. Now, for remediation, most PFAS destruction technologies involve degradation in the solution, but and we have a number of these uh, technologies that have been proposed. But contaminated soils and solids, it's a little bit harder. We'd probably have to extract it by using some sort of solvent and then treat the extracts, which would then kill everything in the soil. And some PFAS are chemically recalcitrant than biological matrices, so it's challenging to remove PFAS from already contaminated food. And what we're finding with the PFAS remediation technology is that they tend to work really well at the bench scale, but scaling to the environmental level is very challenging. Some of these are very energy intensive, and they have the potential to generate toxic byproducts. So for mitigating it in soils, like and it's even harder, so we probably have to model the transport through water and soils, and all the PFAS molecules have different properties, and then you have soils that all have different properties, so it makes it challenging. So some approaches are like maybe you engineer the landscape to prevent transport. Maybe again, you use the wash and you pump and treat, so you get the solvents, and then maybe treat it with electrochemical oxidation. Or maybe you can add some sorbents to the soil to keep the PFAS in the soil so it doesn't transport up into the plants. And maybe other things like soil excavation, absorption, and plasma destruction. So it's hard and requires a lot of investment. But we're not going to get anywhere unless we think of safer replacements. We use these chemicals. We love these chemicals. They're really inexpensive and effective at low doses. So it's really challenging to develop products that are effective and are not toxic, like inert substance. So I think in moving forward with the PFAS, we have to just really assess essential and beneficial use, and then do a full life cycle assessment on when we're creating these chemicals, where is it all going, and making sure that we have proper cleanup if we're going to use these chemicals. So basically that's led to our center where we want to lead. We have a solutions approach for our center. We want to lead development of PFAS measurement standards in food. We want to mitigate PFAS in agricultural and natural products. We want to develop green chemistry guided alternatives based on systems biology. I mentioned that we had the systems tox approaches, but if we could develop safer chemicals by running it through the same suite of assays that we're developing for rather testing whether or not these existing chemicals are toxic, then we start to stop producing these toxic chemicals from the very beginning. You want to advance the state-of-the-art design of rapid, safe, and effective remediation technologies. We'd like to be able to effectively communicate the risk of these chemicals, and we'd like to be a collaboration hub. So we're welcome all who wants to collaborate with us. We have a number of projects in development since we started in 2020. We have about several from the EPA, USDA, CERTUP, the Federal Aviation Authority, NSF, and the Great Lakes Protection Fund, and the Department of Defense. So we have a few of these in development, and we've just started renting a farm and looking at some contaminated cows and biosolid applied land. We're doing a number of different things on this land to help the regulators come up with some thresholds and things for the bioavailability of some of these compounds in different matrices. And with that, I'd like to say thank you so much for listening. And I'd like to thank my funding resources. Our center was funded by the Vice President for research and innovation for MSU and Ag Bio Research. We have a number of different funding sources. And also we have a very talented group of people. And so I encourage you to look at our website and see if there's other people. We would love to collaborate with you. And also say thank you to SCIEX for this kind invitation to present. And with that, I will end.